It is really a great pleasure to be here today. I'm not a direct member of the Heeger school, but as already Serdar mentioned, I am a Santa Barbarian. So I visited Santa Barbara in 1992, and I was at IPOS that Alan was directing, and it was a great pleasure to be there. So it is really an honor for me to be here and celebrate Alan's birthday. First of all, I want to thank all the people that contributed to this work. The synthetic work was done in Trieste. We will use carbon nanotube sponges produced in Roma by Maurizio De Crescenzi. Neurophysiology is in collaboration with Laura Ballerini. TEM and SEM are performed in Trieste at different institutions, and the money comes from mainly from European community, but also from national sources. What Serdar was saying is that my talk is completely different from all the others because it is not related to conjugated polymers, but in a way we can consider nanotubes as extended conjugated polymers in such a way that we can use them for applications in many instances. So when I visited Santa Barbara and I worked in Fred's laboratory, I discovered the activity of fullerenes, and then I promised not to work on carbon nanotubes, but then I got a project funded, so I had to work on carbon nanotubes, and so the things went very well, so we had to continue. And now we get funds for graphene, and so what we can do. They are especially good in covering surfaces, electrodes in particular, for electron transductions. And so the most important issue is that carbon nanotubes and graphene can transfer electrons very easily, and they can be used, for instance, in biological applications, for instance, in the case of synapses, where somehow there is electrical pulses, even though they are of different nature. So what we decided to do about 15 years ago, we decided to study the interactions between neurons and carbon nanotubes. From a morphological point of view, they are very similar, as you can see, even though the scale is different. I mean, neurons, neurites are much bigger in size than carbon nanotubes, but the shape is very similar. And so at a certain point, we decided why we should not put in contact carbon nanotubes and neurons so that we can see how communication between neurons is enhanced in the presence of carbon nanotubes. And so we started with a very simple approach in which we took a glass cover slip, we covered it with functionalized carbon nanotubes, and then we hit the functionalized carbon nanotubes in order to fix the carbon nanotubes on the surface of glass. And then we grew hippocampal neurons on pure carbon nanotubes, and then we let them incubate. What we can see is that the layer of carbon nanotubes is very homogeneous, and neurons grow very normally and very efficiently on carbon nanotubes. What we can see is that the shape of the neurons, the elongations, are natural. So the substrate of carbon nanotubes is a perfect substrate for the growth of neurons. What happens in general, neurons try to communicate electrically with the other neurons. So they elongate their dendrites in order to reach other neurons and start communicating with them. So if we enlarge this image, we can see that this neuron is probably having a synapsis here with this elongation of the other neuron. But what is outstanding here is that the interaction of the nerves with carbon nanotubes is really, really impressive. We can see that nanotubes really become part of the neurons. As you can see here also in another experiment, nanotubes can penetrate inside the cell. So basically what we believe is that there is a tight contact between neurons and carbon nanotubes, which make the two entities to behave as a whole single thing. And so if we look at the other perspectives, and so for instance, if we cut the glass cover slips into transversal sections, so now this is the glass, this is the layer of carbon nanotubes, and this is a section of the neuron, we can see that normally the neuron floats on the layer of carbon nanotubes. But sometimes we see the nanotubes penetrating inside the cell. And so basically, again, this is a confirmation that the two components are really in tight contact. What we do in collaboration with electrophysiologists is so-called patch clamp technique. So the patch clamp technique takes a micropipette which has an electrode inside. This electrode is placed in contact with the main soma of the neuron. And so basically we have an interaction between the electrode and the main soma of the neuron so that the spontaneous activity of the neurons can be recorded by using this electrode, but also the electrode can send pulses to stimulate the activity of the neurons. And so if we record the spontaneous activity of the neurons, this is one single neuron which is recorded in its activity by using this patch clamp technique. These are called postsynaptic currents. These are the electric pulses that the neurons send to the surrounding neurons in a spontaneous way, just to communicate with the other neurons. As you can see, the neurons sitting on carbon nanotubes have a much higher intensity, frequency of sending these signals to the other neurons with respect to the control, which is in this case pure glass. Also, this is another way of measuring the spontaneous activity of the neurons. This is called action potentials. And also in this case, we can see that the activity is much enhanced as compared to the control. So basically, we can see that the increased number of interactions is not due to morphological reasons. So the number of neurites and the elongation of the neurites are very similar. And so there is no such a simple explanation as a higher number of cells or longer dendrites. So what are the reasons for this increased frequency of activity of the neurons? Well, first of all, I have to say that this is not yet completely clear. And I understand this is an audience of people expert in electronic conductance, so maybe you may find a better explanation. One of the reasons could be that we get a direct coupling between the nearby dendritic compartments. This means that if we have the nanotubes can act as electrical wires, and so they can connect either different parts of the same neuron or different neurons. This is a very simple explanation in the sense that it is known that nanotubes are conducting, and so this could be well be the reason. But it is experimentally, this is very difficult to study, and so other experiments aimed at this hypothesis are that if we take a solution of neurons and we place them on a bed of carbon nanotubes, and this bed is prolonged outside the solution, and we can apply an electrical field here, and so if we stimulate here, we wanted to see if these neurons inside the solution get some stimulus. And this is the response that we see after we stimulate this neuron from the outside electrical field. So the electrical field stimulates the neurons in the solution, and we record the activity using this patch clamp technique. This is the response of this neuron to this pulse, And this is very similar to this same reaction of the neuron if we apply the stimulus directly from the patch clamp pipette. So we have a very similar behavior for the electrical stimulation of the neuron. So it looks like the nanotubes can carry out the electrical stimulation directly to the main body of neurons. This is more objective result. We have, in the presence of carbon nanotubes, we have an increased number of synapses. So when nanotubes are present, the neurons recognize each other, and so they can connect in a much better way. And so we have a statistically significant increase of synaptic events. So far, what I have shown was carried out using multi-wall carbon nanotubes, which are considered metallic. But the metallicity, of course, as you can understand, plays a very major role here. So we decided to work with single-wall carbon nanotubes because they can be either metallic or semiconducting, and they can be separated. So we collaborated with Andrea Ferrari in Cambridge, and they covered glasses for us using separated metallic carbon nanotubes, semiconducting, and we compared them with multi-wall carbon nanotubes and the control, which is pure glass. And what we see is that only the metallic nanotubes behave like multi-wall carbon nanotubes in terms of increased number of synapses, whereas the semiconducting carbon nanotubes behave very similar to the control. Also, the spontaneous activity of the neuron, the electrical activity of the neurons is increased in the case of metallic nanotubes, whereas in the case of the semiconducting nanotubes, the behavior is very similar to the control. So the metallicity of the substrate plays a major role here. So at this point, we should think of, there are other materials other than carbon nanotubes that can play this role of increasing the interactions between neurons. And so we decided to use different materials. For instance, indium tin oxide has the same resistivity of carbon nanotubes. Or we used radapeptide, which is a peptide that self-assemble into a nanotube shape. So this will be reminiscent of the shape of carbon nanotubes, but will not be conducting. This is conducting, but does not have the shape of cylindrical shape of carbon nanotubes. In both cases, we did not see any effect. I mean, it was very similar to the behavior on glass. So none of the conductivity alone or the nanotube shape alone can play a role here. And so we also wanted to check what happens with P-dot PSS, which is a subset which is very often used, as we have seen before in the talk of Ole Inganas, which is used to stimulate neurons. And so also in this case, we could produce glasses covered with this substrate, also doped with ethylene glycol to improve the conductivity. And we can see that, in fact, neurons grow very well. Also, glial cells grow very well on this material. However, the activity of the spontaneous activity of the neurons on this substrate is very similar to polyornithine, which is a standard substrate used for these cultures. And so what we understand from these experiments is that neurons and carbon nanotubes like each other very much. Neurons like to grow on carbon nanotubes. And also that there is a sort of electrical exchange between the two entities which favors the interaction between different parts of the neurons. And so at this point, we decided, why don't we try to see whether this material can be used to cure spinal cord injuries, which today cannot find any cure, clinical curing. As you know, when someone has an accident and the spinal cord is displaced, there is no transmission of the electrical signal between the two parts of the spinal cord. This is a very common disease, and it is a very bad disease because it most of the times obliges the people to stay in bed for the entire life. And so we decided what happens actually is that where the lesion occurs, scars start to create. And these scars stop the electrical communications. The scars are insulating, and so they stop the transmission of the electrical signal between the two parts of the lesioned spinal cord. And so we decided to use carbon nanotubes because we thought maybe, since we know that they integrate very well with nervous tissue and they are conducting, maybe we can regenerate the electrical communication between the two parts of the spinal cord. So basically what we plan to do is to lay a bridge between the two parts of the spinal cord in such a way that maybe the two parts can restart communicating. And to do this, we started with a whole slice of a spinal cord. I mean, this slice is a section of the spinal cord, so it's an entire tissue of spinal cord. And so it contains all the elements of the entire tissue. And we wanted to see whether there is an effect of carbon nanotubes also on the entire tissue. And what we can see is that, so the experiment is carried out in this way. Basically, this is laid over a bed of carbon nanotubes. So here we have the interaction of the carbon nanotube layer, and on top we take the recording. So this is a slice of 275 microns in thickness. And basically what we find is that on carbon nanotubes, the number of fibers grow 40% more. So the fibers are much denser in number, but also the length increases by 25%. So the effect of carbon nanotubes can be felt also on entire tissues of spinal cord. Also, we can see that growth cones, which are these elongations that come out of the tissue, grow more distant on carbon nanotubes. And so now the main experiment comes. We can take two spinal cord slices. We place it either on glass or on carbon nanotubes. And we can see whether the two slices restart communicating or not. When we did this work on planar carbon nanotubes, we could not see any effect. So we had to change strategy, and we used these carbon nanotubes, which are produced in Roma by Maurizio De Crescenzi. They have relatively high conductivity, and they are very big carbon nanotubes, but they are especially in the form of sponge. So it's a relatively spongy, elastic material, which can be really treated like a very easy material. And so this is the shape of carbon nanotubes that we have used. You can see the three-dimensionality, and neurons grow attached to these fibers in a very, very tight contact, very similar to what we have seen before. So now, if we do a three-dimensional reconstruction of the sponge, this is the glass where the sponge is attached, and these are the nanotubes. This is the empty sponge, and this is the filled with neurons. And so we can see that the neurons grow in 3 dimensions, and they grow very well. So also this material is a very permissive material for the growth of neurons. And here we can see this is the overimposition of several planes going from top to bottom. And when we place neurons, we can see that the neurons really grow very efficiently in 3 dimensions. Okay, so now the experiment comes. We have the two slices. We can either stimulate one slice and record on the other, or record the spontaneous activity of the two slices. Well, first of all, the morphology. We have the two slices. This is spinal cord explants. The two slices on the control. You can see that between the two slices, there is no really tight contact. The fibers grow. The fibers are very thick in diameter, very strong, but they don't try to go towards the other one. When we place the two spinal cord explants on carbon nanotubes on the 3D sponge, we can see that here all the fibers grow much thinner than here, but that between the two slices we have a very thick layer of carbon nanotubes. So now the electrophysiology. Basically, when we have the two slices, we see this is the spontaneous activities. These are the electrical signals that each spinal cord explant sends spontaneously. And we can see that they are not coordinated at all. So each slice, it's completely independent. If we try to stimulate one of the two slices, for instance, this one, with continuous regular pulses, we see that the other one doesn't respond at all. So this is an enlargement. You can see these are the pulses. So this is the first slice responds in this way, but the other one completely ignores the stimuli, the electrical stimuli. In the case of carbon nanotubes, we see that already at the stage of spontaneous activity, we have a perfect correspondence of the signals. And if you stimulate one slice with regular pulses, then we can see that the other one responds very efficiently. So this means that the two slices now are completely electrically connected. And this is, of course, very promising in terms of what we want to explore, because now what we are doing, we are placing the device in the spinal cord of the lesioned animal and see whether this approach will work or not. So this is the end of my story. Again, congratulations to Alan for his 80th birthday, and thank you for your attention.